So let's begin Chapter 4 on the tissues. When you were formed, you were the product of your mother's egg and your dad's sperm that came together to be a zygote. And that zygote cell started dividing very quickly, cleaving really, not completely dividing, but growing from a mass of 1 cell to 2 cells to 4 cells to 8 cells and so on until you went from being a zygote to a morula to a blastula and then a blastocyst, which is hollowed out. And so on the screen right here, you can actually see that this is a blastocyst where it's hollowed out in the middle. It's at that blastocyst stage that you begin to differentiate. No longer are all the cells identical, but because of DNA and hormonal direction, we have 3 distinctive germ layers or beginning layers: the endoderm, the mesoderm, and the ectoderm. And from each one of these germ layers arises our 4 tissue types. So let's just think about it a minute. Epithelial is a type of tissue. It covers and lines. So epithelial is going to come from the ectoderm, from the endoderm, but epithelium also makes up glands, which would be in the middle, so it comes from the mesoderm. It comes from all 3 because it covers, it lines, and it makes up glands. Now let's think about muscle. Muscle is deep to epithelium. It's in the middle, so it's going to come from the mesoderm. Most connective tissue is going to be binding one thing to another, so it's going to be in the middle, mesoderm. And then the nervous tissue is going to come from the ectoderm. And the best way I can help you remember that is that when you start developing and you go from this blastocyst into an embryonic stage, it's like the nervous system, which is the dorsal cavity, is kind of bent and curved over, and then it just arises up. So in that very early stage, the dorsal cavity would be, the bony back, the outside, and then it's going to, as it develops, move on up and become the cranial cavity and the spinal cavity. So it's going to be on the outside and it moves up. So that comes from the ectoderm. So maybe this will help you understand the notes a little bit better when I say what germ layer forms this type of tissue. So we already know from A&P from Chapter 1 that epithelial, connective, muscle, and nervous are the 4 types of tissue. So let's get a pretty quick overview, and then we're going to look at each type of epithelial tissue, each type of connective, each type of muscle, and each type of nervous with more detail. So what does epithelial tissue do? Well, it covers surfaces and it lines cavities. And I realize my I should be after my T in cavities. It covers surfaces and it lines cavities. So it's going to line hollow organs, like the inside of your stomach is going to be epithelium. The inside of your heart is going to be epithelium. But it also makes up glands. So this is where it would dip down into the middle and be glandular in nature. So that's coming from a little bit deeper than the epithelium that covers and lines. So what germ layers does epithelium come from? Well, since it covers, ecto; since it lines, endo; and since it's glandular, mesoderm. It comes from all 3. Epithelial tissue can regenerate very, very well. It's constant. Since it is covering surfaces and protecting, as it sloughs off, it has to be replaced. So it has constant and excellent regeneration. Let's get some beginning information on connective tissues. Connective tissues do just what they say, is they connect. They support, they protect, and they connect or bind organs. So you're going to find them supporting epithelia. You're going to find them protecting epithelia. You're going to find them binding epithelia or connecting epithelia to muscle underneath. One special type of connective tissue is adipose, and so it stores fat. And another type of connective tissue is the blood, and so there are white blood cells in the blood, and that's going to give us immunity. Which germ layer does connective tissue come from? The middle one, or mesoderm. And whether connective tissue regenerates well or not depends on the blood supply. Most connective tissue does regenerate, but the cartilage is avascular, so it doesn't regenerate as well as others. And tendons and ligaments, which is made of fibrous connective tissue, has a scanty blood supply, so it doesn't regenerate as well. So it has a lot to do with the blood supply and how quickly we can provide nutrients and clot off the area and other factors such as that. The third type of connective tissue is muscle, and of course we know muscle provides movement, but it also supplies force. I can push on a wall and not move it, but my muscles are still providing the force at which I'm exerting on the wall. So not only movement but force as well. Remember, it develops from the mesoderm since it's in the middle, and it has poor or no regeneration. There are some satellite cells that can regenerate, some support cells, but the norm is that muscle cell does not regenerate. The fourth type of connective tissue is nervous tissue, and nervous tissue is going to help maintain homeostasis by detecting and responding to stimuli through nerve impulses. So there are receptors throughout your body that monitor when things are out of balance. When a message is received that we're out of balance, we send an impulse to a control center like the brain or the spinal cord. We integrate, figure out what to do, and send a response back to either muscles or glands. And so nerve tissue coordinates all the body's activities. We looked at how it comes from the ectoderm, and most nerve cells do not regenerate. So we have nervous tissue, muscle tissue, epithelial tissue, and connective tissue. So we're going to start with the epithelial tissue and look at some characteristics and then learn the different types of epithelium. Epithelial tissues are closely packed. There's very little intracellular space between epithelial cells because we're almost building a wall around something if they cover in line. So they're closely packed, very little extracellular space. Since they're closely packed, they adhere to each other. They have tight junctions and desmosomes, and sometimes when you try to lift one epithelial cell off, you'll get a whole sheet because they are held together by those tight junctions and those desmosomes. So in lab, when you tried to scrape your epithelial cells off of your cheek cells, you probably may have seen cells stacked on top of cells instead of seeing a lot of individual cells. They come off in continuous sheets. Epithelial cells have what we call polarity. They have one end called the apical end that is facing out to the cavity they're lining, or the surface they're covering. They're out to the air. So if this is your epithelial cell, then this is sitting on another connective tissue basement membrane, and this is the edge that's to the free surface. So this is the apical edge. Some apical edges have finger-like extensions. You should already know what those are. They are microvilli, and then others have hair-like extensions that move. Those are cilia. So we'll find those on the apical edge, the edge that is into the space. The other surface is called the basal surface, and it's going to be on a very thin basement membrane. You can see down here that this is a very thin basement membrane. This would be connective tissue underneath. This would be epithelial tissue, and see, there's the space. So that's the apical end. This is the basal end. It has poles. That's why we say it has polarity. So the basal surface is attached to a basement membrane, and it acts as a selective filter between the epithelia and the underlying connective tissue. Epithelial tissue is avascular, so in order for these cells to receive their nourishment, they have to get it from the connective tissue underneath. So that basement membrane in between that's under the basal surface acts as a filter. That basement membrane has a non-cellular adhesive sheet of glycoproteins called the basal lamina, and then it has a network of collagen fibers called the reticular lamenta. And so you can see a picture here of the lamina. The basal lamina would be the glue in between, and then the fibers would be the reticular lamina. And so it's kind of like a layer that connects epithelia to whatever's underneath it. Most of the time it's connective tissue. So it's kind of like a glue and fibers that hold the epithelia to the underlying tissues. And if you notice, number 4 says it's supported by connective tissue. Since it is avascular, it's going to receive its blood supply or its nutrients from the blood, oxygen by diffusion from the connective tissues. Also get rid of waste the same way. So that should tell you that epithelial tissue can never be very thick if it's relying on structures under it or around it to provide its nourishment. It has a very good nerve supply. Your epithelia is one of the main places for some of your receptors for the nervous system that monitor what goes on. The only two responses you really have as a person is to move or not move, or secrete or not secrete. And since glands are part of epithelium, you would have a nerve supply that regulates the secretion from those cells. Epithelial tissue has a very high rate of mitotic division. They are covering surfaces and lining surfaces. Substances are moving across them, and so they're an area of high wear and tear. They slough off, they shed, so they have to be replaced very quickly. So what are some of the functions of epithelia as they cover, line, and make up glands? They protect. They act as filters sometimes. They lubricate. That would be a glandular part, releasing mucus. They secrete substances like hormones. They can be the membrane across which we absorb our nutrients. We can get rid of waste out of epithelia. That's excrete. There are lots of sensory receptors there for pain and touch and pressure. So it is a sensory perception tissue. So how do we name epithelia? Well, we name epithelia by the layers that are of the layers of cells in the epithelial tissue, and by the shape of the cells. So let's look at the arrangement of layers first, and then we'll look at the cell shape. If you only have one layer of one type of epithelial cell, you call that tissue simple. If you have two or more layers of one type, then you say it's stratified. There are some tissues that look like they're more than one layer. Look at this tissue. It has nuclei all at these different layers, but if you look, you can trace every one of those cells down to the basement membrane. You might not see where it comes out the apical surface, but they're all sitting on the basement membrane. So it really is one layer, but it looks like it's many, so we say it's pseudo-stratified, falsely stratified. So what are the shapes of cells? Well, if we have a cell that's short and squatty, it's squamous. They kind of fit together like puzzle pieces. If you have one that's about as tall as it is wide, like a cube, it's cuboidal. If you have one that's taller than it is wide, like the column on the front of a porch, it's columnar. And if you have one that changes its shape based on the tissue it makes up, then we call it transitional. And the best example I can give you of transitional is if I'm a cuboidal epithelial cell and I'm lining the lumen of the urinary bladder, and my urinary bladder fills up with urine and it stretches the tissue, it could stretch a cuboidal cell all out and flatten it so it looks squamous. So the shape is dependent upon the tissue that it makes up. So as it's stretched, it changes its shape. It's transitional. So let's look at some simple epithelial tissue. In other words, what we're going to do is we're going to put simple with squamous, simple with cuboidal, and simple with columnar. So this would be one layer of flat scale-like cells, so it's simple squamous. You find simple squamous where you have diffusion and filtration surfaces, like in your small intestine. You're going to have simple squamous so you can diffuse products that you're absorbing across. This would be one layer of cube-like cells, so it would be simple cuboidal. You find these in areas where you need secretion and absorption surfaces as well. This would be simple columnar. Some of these are going to be non-ciliated. That means at their apical end they don't have any hair-like extensions. Some of them that are non-ciliated are going to have the microvilli at the top of them to increase surface area, like in your small intestine where you want to absorb. You have microvilli to increase surface area. And usually along with the microvilli, non-ciliated, simple columnar cells, you will have a cell that looks like this called a goblet cell. A goblet is kind of like a short wine glass, so it's big at the top and then it goes down to the stem. And so they named it because they said it looked like a goblet. But its job is to secrete mucus. So where you find non-ciliated simple columnar cells, you usually find these goblet cells. I remember what a goblet cells produce because I would never want somebody to serve me a goblet of mucus. Another extension we see sometimes of the simple columnar cells is that it's ciliated. You'll see cilia at the apical end, and that's to move mucus or something along the surface of the cells. So let's look at this epithelial tissue. The trick to finding epithelial tissue is to always find an open space. So this is an open space, and what is covering or lining this open space? A very, very thin line. It's so thin we have to know it's simple, and we can only see a nucleus every once in a while, so the cells have to be short and squished, squatty. So this would be simple squamous that lines this space. Look at this. This is the space, so these are the cells that line that space. You can't really see the line in between them, but you can imagine that there's a line in between them and that they are going to be cuboidal. See, there's one, and there's one, and there's one, one. Here's a perfect one. It's as tall as it is wide, one central nucleus. This would be the lumen that it is lining. This is simple cuboidal. Here's another layer of simple cuboidal, another layer of simple cuboidal. It kind of reminds me of wagon wheels. Here's our space right here in the middle, so that means this must be epithelia. It's one layer of taller than wide cells. You can see the nuclei are all at one level. That's the basement membrane where they're sitting, so this is the apical surface. So this would be one simple columnar layer, and this would be another simple columnar layer. Sometimes we have pseudostratified columnar where it looks like it's more than one layer, but if we could see each cell, they go all the way down to the basement membrane. Notice at the apical edge we have cilia, so this would be ciliated pseudostratified columnar, and these little funny looking cells found in between them, those are goblet cells that are going to be producing mucus, so the cilia is going to help to move the mucus along. So how about stratified epithelium, more than one layer? We find stratified squamous, superficial squamous, and deep cuboidal or columnar. Let me explain that. When I look at the stratified squamous tissue, I'm always going to name the stratified after the apical edge. So up here at the apical edge, they look short and squatty. But if you go deep, they really look cuboidal down here. We always name them by what's at the apical edge. So since this is more than one layer, it is stratified squamous. Sometimes stratified squamous is keratinized. That means that as the cells move towards the surface, their cytoplasm is replaced with keratin, and keratin is a waterproofing protein. Your skin is keratinized stratified squamous epithelium, so that by the time you get to the outer surface, really these outer apical cells are dead. They're filled with keratin, and they simply make a waterproof barrier. So your living cells are down here in your bottom layers, and as we move up, we fill with keratin and they die. So that's why when you get in the bathtub and all that old skin cells slough off, it doesn't hurt because those cells are dead. It forms a very waterproof barrier between us and inside and outside world. Sometimes we have non-keratinized stratified epithelium. Think about inside your mouth. You wouldn't want a waterproof layer inside your mouth and kind of feel like you had grease coating your mouth. So these are non-keratinized they're still going to slough off. They're still going to die as they move up towards the apical end, but they don't have the keratin in them. There are areas in the body where you have stratified columnar and stratified cuboidal, but they're rare. You're going to find them providing protection, limited secretion, and absorption. This would be a pseudostratified epithelium. So you can see that they kind of look columnar here, and then as we move to the top, they look flat and scale-like. So what about that transitional epithelium that I talked about a while ago? When the organ that they're making up is relaxed, then they look one shape, and then when the organ fills up or stretches, they look another shape. You really couldn't look at a tissue and say, oh, that's transitional. You'd have to see it in its relaxed state and its distended state to really tell that. But the urinary bladder is a good place to see transitional epithelium. So what about glandular epithelium? Glandular epithelia lies deep to covering and lining epithelia, so that's why it comes from the mesoderm, and it's going to secrete. A gland, by definition, is a cell or a group of cells that secrete substances into ducts, onto surfaces, or into the blood. The cells that are making that substance, they don't need that substance. It's needed somewhere else. So if we are an endocrine gland, we are going to secrete our substances right into the bloodstream most of the time by diffusion, and it's going to travel to a target organ in the blood and affect that organ maybe, all the way across the body. From where it was produced. An exocrine gland is going to secrete its substances through a series of little ducts or tubes, not quack ducts, but little tubes. So you're going to have a tube coming from the gland into another organ. For example, your pancreas is endocrine and exocrine. It's going to make digestive enzymes that it releases through a series of little ducts, and it empties into the small intestine to break down food. But the pancreas is also endocrine in that it makes the hormones glucagon and insulin that it dumps into the bloodstream to go to places in the body to either take sugar out of storage like the liver or to put it back into storage. So your book has a lot of classifications of exocrine glands. I'm not going to be that particular. I want you to know that you can have a unicellular exocrine gland. That would be like that goblet cell, that one cell that makes mucus and releases it onto a surface. Or you can have multicellular, where you have a distinct structure like the pancreas. You can have exocrine glands based on how they release their secretion. Merocrine exocrine glands are probably the most common type, and what happens is that the little vesicle, a secretory vesicle, goes to the edge and releases its substance into a duct, and then that duct leads into another organ. Another kind of exocrine gland is called a holocrine exocrine gland. If you think about the Holocaust, a lot of Jewish people were killed, so holo meaning death. So in this case, the cell is going to die, and the edges slough off and they release its secretion. Some people think that maybe the release of milk could be this type, or it could be apocrine, where the apical end just pinches off. So if the apical end pinches off, it's apocrine. If the cell dies and releases its substance, it's holocrine. And most cells are going to release by a little vesicle, exocytosis, into a series of ducts. So you need to know the three different functional classifications of exocrine glands. So now we are to connective tissues. Okay, so connective tissues are the most abundant and the most widely distributed. They vary the most, but they all have three things in common with each other. There are very few non-touching cells. Remember, epithelial is stacked one on another. They form barriers and boundaries, but the cells in connective tissue are spread out, and in between these spread out cells is an extracellular matrix. So if you look at this picture of just a typical connective tissue, you can see different kinds of cells, and they're just spread out. They're not stacked one after another. And so this stuff that's in between the cells is called the matrix, and that's where the connective tissues vary. In bone, that matrix is hard calcium phosphate salts. In blood, that matrix is plasma. It's actually almost water. In cartilage, that matrix is almost a plastic-like substance. So we vary from one connective tissue to another by varying the matrix that's in between the cells. Connective tissues function to bind and support. Some of them actually protect. Fat is a type of connective tissue that insulates. Blood is a type of connective tissue that transports. They don't line anything except for joint cavities. They have a very good blood supply, except cartilage is avascular, and dense regular connective tissues and irregular connective tissues, the blood supply is scanty there. So that's going to have a little bit to do with their regeneration ability. So what is this ground substance, or this matrix that's found between the cells of connective tissues? It's a homogeneous, that means that it's the same all the way through, extracellular substance that ranges from liquid to solid. No matter whether it's a liquid or a solid, it's usually a good diffusion medium so that we can diffuse substances across or through connective tissues. It's made of interstitial fluid, adhesion molecules, and proteoglycans that trap water to give the tissue more turgor or make it more stiff. And so this matrix is kind of like the glue found between the cells, but the glue varies from very hard in bone to very liquid in blood. Another characteristic that all connective tissues have is in the matrix you're going to find fibers, and there are three possible kinds of fibers that are going to strengthen, support this connective tissue. The most common, the most abundant protein in the whole body is collagen. It's tough and it has tensile strength. What I mean by tensile strength is it has the ability to give a little bit before breaking. In Mississippi, when we have bad weather, pine trees, they don't have a lot of tensile strength. They're either going to snap or it's just going to lay them down with their roots showing. But a tree like a willow tree is going to give a little bit with the wind. This is going to kind of stretch and then pop back up. And that's what we mean by tensile strength. So collagen gives our tissues the ability to stretch a little bit, still support, but stretch before breaking. We also have elastic fibers. These are the ones that really give us elasticity, where we can stretch a lot and then recoil. And then the last kind of fiber you really only see in lymphatic structures. It's called reticular fiber. It's thinner than collagen fibers, and it is the framework of soft organs like the spleen, which is part of the lymphatic system. So all connective tissues have cells that are spread out, a matrix that's in between the cells, and in the matrix you find fibers. So let's talk about some of those cells that you could possibly find in connective tissues. Not that you're going to find these in all connective tissues, but then in different types. One cell that you find in most connective tissues is a fibroblast. A blast, anything with blast means a maker. So this particular cell is going to make fibers. In cartilage, which is a connective tissue, the cells that are there are called chondroblasts, and they're going to make chondrocytes, which are cartilage cells. Bone is another connective tissue, and you'll see osteoblasts that are in bone. Osteoblasts are going to make bone cell and bone tissue. You have hematopoietic stem cells. These are the beginning cells that you find in blood. Your white blood cells, red blood cells, and platelets are all going to arise from these stem cells in blood. Other cells we might see associated with connective tissue are adipocytes. You're going to find these fat cells in adipose stores around the body. That's how we're going to store fat. Then you're going to see white blood cells. They actually originate in the bone marrow, travel or released in the blood, and then can leave the blood and go all over the body, giving us immunity. So let's look at the different kinds of connective tissues. One type of connective tissue is called loose connective. If you look at this picture, you can see that the fibers are very loose. It looks very like it has a lot of space. It's very airy So this is areolar loose connective. You have all three kinds of fibers, but few in number with increased viscous matrix. So the matrix is going to be thicker than water. It's going to be almost gel-like, and that's hyaluronic acid that makes it have this viscous matrix. You're going to see cells in this areolar tissue, like right here and right here and right here. Those would be fibroblasts that makes the fibers. You also see macrophages, which is a kind of white blood cell which would be giving us immunity. Areolar connective tissue is the most widely distributed connective tissue. It's what gives an organ guts consistency. You will also find it under every epithelial tissue because it is the tissue that makes up the basement membrane. So here would be epithelial tissue, and this would be areolar connective tissue making up the basement membrane. Another kind of connective tissue we see is adipose. Adipose is made up of adipocytes that store triglycerides, and it's usually found under or around areolar connective. It's a good insulator, and it stores energy. And all these little white marshmallow-like structures you're seeing are the fat or adipose, and they've pushed everything else in the cell to the side. Another type of reticular connective tissues only have reticular fibers, and you find them in lymphatic structures. So let's talk about dense connective. There's more fibers and less cells. Look at this one. All of those wavy things you see in the background, all of those are connective. All of those are fibers, and they're white collagen fibers, and they're running one after another in a regular pattern. So we call that dense regular, and we find it in places where we we need like a strap-like connection, like with ligaments or with tendons. So another type of dense connective tissue is irregular. You can see with this second picture that the fibers aren't all running parallel and in the same direction, but they're going every different directions, and this is where you need strength in several directions, like with the valves in your heart. Another kind of dense connective tissue is elastic connective, and you see here that the elastic fibers are running very close to each other, and you're going to find this in the area where you need to stretch and recoil, like within arteries. Another kind of connective tissue is cartilage. Cartilage withstands tension and compression because it has lots of collagen and elastic fibers in it. It's got a rubbery chondroitin matrix and chondrocytes, which are cartilage cells, in lacunae. It is an avascular connective tissue, so it's never going to be very thick. It has it's nerve-free except in the membrane that surrounds the tissue, and avascular-free. So in this connective tissue cartilage, here you would see no blood vessels or nerves, but if there was a membrane around the cartilage, that's where you would find the blood vessels that feed the cartilage, and that's where you would find nerves to monitor the environment and send sensory information. Now notice that this is kind of almost like a plastic-like matrix, and these are the chondrocytes, and they're in little spaces called lacunae. You're going to see lacuna or lacunae. You're going to see these in bone too, so they're all like little cavities that house the cartilage cells. So how is that cartilage cell going to be fed? By diffusion from the membrane that surrounds it. The membrane that surrounds it is called the perichondrium. Peri meaning around the perimeter or outside of the cartilage. So we have three different kinds of cartilage. We have hyaline cartilage, which is the most abundant. It is the weakest. We find it attaching the ribs to the sternum. It does give flexibility. It does reduce friction. You'll find it at the ends of long bones in a joint, so where your arm bones come together at your elbow joint or at your shoulder joint, those bones will be covered with hyaline cartilage, so it gives us almost a friction-free surface for those bones to slide across. A second type of cartilage is called fibrocartilage. This one is the strongest. You can definitely see the collagen fibers there. The collagen fibers were so fine in hyaline cartilage you can't see them, but you definitely can see the fibers. You still have your chondrocytes in the lacuna. You find that your intervertebral discs are made of fibrocartilage. Think about your stacks of backbones, one on top of another, with your very heavy skull at the top. So they have to be strong and not be compressed. And then the last kind of cartilage is elastic cartilage, and so it has lots of elastic fibers in it. So it's going to be able to stretch and recoil very well. If you take your ear and you pull it anteriorly and lay it down in front and then let it go, that's because you have elastic cartilage in that pinnae or outer part of your ear. So you can see the elastic fibers, and you still can see your cartilage cells in the lacuna. Bone is a connective tissue. Everybody wants to list bone or skeleton as a separate tissue, but it is truly a connective tissue. Another name for it is osseous tissue. I mean, do you think about the old song, this bone's connected to that bone? So connective, bone is connective. It has lots of collagen fibers, and it has calcified salts, calcium phosphate, that makes the matrix very hard. The cells are called osteocytes, and they're going to be found in lacunae, arranged in concentric circles, that gives the bone tensile strength, allows it to bend just a little bit without popping, and hardness. So the salts give it the hardness, and the fibers give it the tensile strength. So right here in the center of this osteon unit, that's where the blood vessels are, and these little dark spots, those are the lacunae with the osteocytes in it, and then the little radiating canals going out from the central blood canal, those are canaliculi. So the cells are fed through these little canals coming and radiating out. So it almost looks like a tree trunk in its arrangement. You have these different little rings that are called lamella, and they stack one inside of each other, concentric circle, and that makes for a very, very strong structure. Osteon units. Your liquid connective tissue is blood. That means that this matrix in between the red blood cells, white blood cells, and platelets is almost water. It's called plasma. It has things dissolved in it and things in colloid in it, but that plasma is 75 to 80% water, so this is a liquid connective tissue. There are fibers here, you just can't see them. They're only going to become activated when blood clots. The three kinds of cells that you see in blood, the most numerous are red blood cells, then it's going to be platelets, and then white blood cells are the least most common. So red blood cells, white blood cells, and platelets. So there is a table in your book that does a comparison of the different connective tissues. There you see connective tissue loose and dense. Then you see cartilage, the three types. Then you see bone. And actually there are two types of bone tissue, compact and spongy, which we'll talk about in the skeletal system. The liquid connective tissue, blood. So that leads us to the third tissue, muscles. Muscle cells are arranged in long, or some of the muscle cells are arranged in long cylindrical structures called fibers. So when we say muscle cells, sometimes we use muscle fiber. This entire length, and it's actually probably longer than that, is one skeletal muscle cell. It's the result of several embryonic cells fusing together to give you a multinucleated long fiber of a cell. If you notice, these are fibers too, but they're short and they're branched, so this cell starts right there and ends right there. This is a cardiac muscle cell or fiber. This is smooth muscle. It doesn't have the striations like the other two do. They're tapered cells that stack one on top of each other. It almost reminds you of epithelial tissue, but it's fat in the middle, and it's got spindles on each end where it goes out to points. That's called a spindle-shaped cell. So, three types of muscle. Skeletal muscle is voluntary muscle. That means that you control it. It provides movement for the skeleton and the skin. That's what gives you facial expressions. They're long striated cells with many peripheral nuclei. If we compare skeletal muscle to cardiac muscle is also striated, but it has a central nucleus. It's involuntary. It's going to cause the heart to pump or contract. And the cells branch, and they only have one central nucleus. If we look at smooth muscle, we have no striations. They're also involuntary like cardiac. They're going to cause hollow organs to constrict like your stomach and your small intestine, and they are non-striated. They are spindle-shaped cells with a central nucleus. So both cardiac and smooth have a central nucleus. Both skeletal and cardiac have striations. Both cardiac and smooth are involuntary and have a central nucleus. So they have some characteristics they share with each other. This would be skeletal, this would be cardiac, and this would be smooth. To me, smooth muscle doesn't even look like a muscle at all. It looks more like epithelial. And that brings us to our last tissue type, which is nervous tissue. In nervous tissue, there's really basically two kinds of cells. You have your very large cell that carries impulses called a neuron, and then you have lots of these little bitty cells in the background called neuroglial cells that are support cells. So this is the neuron. This is a big neuron. You have the cell body, and then you have extensions that are dendrites bringing the impulse into the cell body, and then a long axon bringing it away from the cell body. So you have dendrites, the cell body, and the axons, and they convert stimuli to nerve impulses that they send to control centers like the brain and the spinal cord to figure out or integrate what to do and send a message back. Neuroglial cells are much more numerous, but they're a lot smaller. They're the support cells. They don't conduct impulses, but they support the neurons. The neurons could not do their job without them. We'll talk about these two types of cell in more detail when we get to the nervous system. So, when tissue is damaged, how is it repaired? Well, we want tissues to regenerate. We want to have perfect repair, but that's not the case a lot of times. Sometimes, especially as we get older and nutrients aren't there, our germ cells aren't there, sometimes instead of giving new regeneration of tissue, we get scar tissue or fibrosis. So what we want is regeneration, but sometimes what we get is scarring. So what happens when you damage a tissue? So we damage a tissue, and one of the first steps into the repair process is called inflammation. What happens is that the tissue becomes red, hot, swollen, and painful, and that's because you release histamine. Histamine is a vasodilator, so it's going to make your blood vessels in your area bigger and more permeable. So if you have more blood in the area, of course it's going to be red and hot, and if it's more permeable, more fluid is going to come out, and so it's going to be swollen, and that extra fluid is going to push on pain receptors and cause the pain. So this is the beginning, though, inflammation of repair, because white blood cells will come into the area and start fighting. Chemicals that you need to repair with will come in. You'll form a clot, so you'll stop losing blood. The second step is that you're going to organize. I kind of think about it as laying down a pattern that you're going to grow your new tissue over. You lay down granulation tissue, and then the hopefully new tissue will regenerate over that. So it's kind of like setting down a pattern. It says the clot is replaced by granulation tissue, which restores the vascular supply. Fibroblasts produce collagen fibers to bridge the gap, so you're laying down that pattern. White blood cells called macrophages phagocytize any debris or pathogens that might be there, and then the epithelial cells start regenerating very, very quickly. So the third step is regeneration and fibrosis. Either you're going to completely heal over that granulation tissue pattern, or connective tissues are going to come in, and scar tissue is going to take the place of what was a functioning tissue before. So the steps for repair are inflammation, organization, regeneration, and fibrosis. So what conditions can affect how well we repair, whether we regenerate or we have scar tissue? Our nutrition. If we don't have the proper nutrients, we can't regenerate. Blood supply. If it's a tissue where there's not a good blood supply, then we'll lean more towards the scarring and less to the regeneration. Age. The older we get, the less we regenerate and the more we scar. Infections. If your body is having to deal with an infection from a pathogen, it's going to put all of its efforts there, and so the repair process will either be slowed down or impaired. And then, of course, different tissues regenerate at different rates. If you break a bone has a very good blood supply. It's actually going to regenerate and repair faster than maybe the tendons and the ligaments of the joint that the bone is connected to, because they have a scanty blood supply. Membranes. When we put epithelia on a connective tissue, we call that a membrane. So epithelia and the connective tissue under it is a membrane. We have 4 kinds of membranes in the body. In Chapter 1, we learned that there are closed cavities and open cavities. You know that closed cavities are lined with serous membranes. Closed cavities are lined with serous membranes. That word serous means watery like. So they are lined with parietal serous membranes, and then the organs that are in the closed cavities are covered with visceral serous membrane, and there's a watery serous fluid between them. So let's just think about your heart. As your heart contracts and relaxes, when that outer visceral pericardium rubs up against the parietal pericardium, we want it to be friction free so we don't have inflammation. So that's what the serous fluid is for. Open cavities are lined with mucous membranes. Think about when you open your mouth, you let pathogens in from the outside, so that mucus will catch the pathogen so your immune system can take care of it. Mucus is on a mucous membrane. Synovial membranes line joint cavities, and these are the only ones that lack the epithelia. It's not really a true membrane, but since it's lining that joint cavity or that synovial cavity, they put it in with the other membranes. So it only has connective, it doesn't have epithelial. And then the only dry membrane is going to be skin. Doesn't need the lubrication because it's not 2 surfaces rubbing together, but is your outer surface. It is keratinized, stratified squamous epithelial over irregular thick connective areolar. The epithelial is going to be known as epidermis. The connective tissue is dermis. Epidermis and dermis. And that is Chapter 4 on the tissues.